In this lecture, we're going to look at the hormones that use intracellular receptors, so those that are lipophilic and therefore need a transport protein to be moved throughout the body and the blood, and then once they get to the tissue where the receptors are, though, they can freely cross the cell membrane, go into the cell, bind to the receptor. The receptors are either going to be in the cytoplasm or in the nucleus itself. And so I'm going to start out by going through each of these types, the steroid hormones, thyroid hormones, eicosanoids, and then we'll talk about a bit more about how they function. So to start out, we're going to look at the steroid nucleus. Basically, what this is the general shape that is at the heart of all steroid hormones. And it's important to notice that it has this 4 carbon rings associated with it, A, B, C, and D. And then it has these areas where we typically see different structures bound to it. So those are represented by those little dots in A. And then in B, it shows the general configuration that we tend to see with these hormones. And we're going to, in the next few slides, we're going to look at some specific examples of this. But you'll notice as we go through the slides, they're all going to have a very typical shape of this 4 carbon circles and then different oxygens or other hydroxyl groups or other groups attached to it, depending on what hormone you're looking at. So we're going to start looking at progesterone, and you probably are familiar with progesterone as it is important in the reproductive hormones, but it's also the precursor for all of these steroid hormones. So as you can see here, cholesterol is the basis for creating for these hormones to come from, and we lump them all together in what's called the P450 enzyme family. So I highlighted in red here, and in the next slide all the spots where we see these different variations of an enzyme of P450, and that the enzymes are really important and generally the limiting factors when it comes to converting into these hormones. What's really important here is to see that there are several steps between going from cholesterol to progesterone, and then that P450 plays a very important role in that. I'm not so concerned with this or the next few slides on you knowing every little detail or all of the little chemical changes that occur. It's the general idea that you go from cholesterol to progesterone using these enzymes, and then the progesterone is the basis for the other steroid hormones. Okay. So here, as you can see, corticosteroids are created from this overlying structure of progesterone. And I mentioned, I highlighted in red all of the places where that P450 is an important enzyme in undergoing this change from progesterone to cortisol or aldosterone, depending on which hormone you're talking about. Now, notice that they're not all the same enzyme. They're a variation of that P450. It's a cleaving enzyme, but they have different, slightly different shapes to them. They're just under that same family. And again, I'm not so much interested in you memorizing each step, but recognizing that there are multiple ways that we can get from progesterone to cortisol or progesterone to aldosterone. And depending on the organ and the process, that will dictate which route is taken. Now, the production of what are known as androgens, the most common of which you're probably familiar with as testosterone, occurs in 2 different places. We've got in the adrenal cortex, in the adrenal cortex, and in one layer of the adrenal cortex, we have androgen synthesis, and usually what you end up with is the DHEA, and so that step of that pathway, the pathway that is most frequently followed in the adrenal gland, is that delta 5 pathway, which is basically categorizing a different way that the chemical changes are occurring. You still have the P450 involved, but it's going from, instead of going from progesterone, you're going from pregnenolone, which was that intermediary on the earlier slide, to production of progesterone. So instead of having progesterone as an intermediary, you have pregnenolone, and then you go through the steps to create that DHEA, and that is an active androgen that is again synthesized by the adrenal cortex. The other more commonly known and understood production of testosterone is going to occur in the testes. And this utilizes the delta 4 pathway, where you go from progesterone through the different stages to get to testosterone and to something known as DHT, which is another active hormone that is an important sex steroid hormone. So again, this diagram highlights the different ways that you get androgens in different places, and we generally refer to it as either the delta 5 or the delta 4 pathway. Okay. Now, we do see some of this in the adrenal gland, but mostly we see this in the ovaries. So from that DHEA, we get production of either the testosterone and then the intermediary of estradiol, which leads into estriol, and those are active estrogens. You've probably heard of estrogen as this kind of catch-all term, but it's really representative of these three different hormones: estrone, estradiol, and estriol. We will talk in more detail about their importance later in the semester, but here you can see that either testosterone or DHEA act as the intermediaries to produce these. And I've highlighted up in the upper right part of the image aromatase. Now, we're going to come back to that enzyme again. It's a very important enzyme that allows for the production of estrogens, both from testosterone and from DHEA, to basically get the active compounds, the active hormones. Now, I've mentioned this before, but I want to talk about it in more detail, that because these hormones are lipophilic and able to freely cross the cell membrane, they don't easily dissolve in blood, and they're not easily transported in blood. And therefore, they have carrier proteins that help to carry them through the blood and allow them to reach their target tissue. Now, I listed the different types of binding proteins on the left. You can read those yourself, and we'll talk about them again. An important thing to realize is that some of these will be free. Some of these hormones will be free, and once they reach the tissue where the receptors are, they have to be released from their carrier proteins. And so this occurs as a consequence of concentration gradients. So once those hormones reach the tissue that has the receptors and start being kind of leaving the blood, that creates this decreasing concentration, which causes the further release of the hormones at the target tissue. So just by way of the concentration of these hormones in the blood and the availability of the messengers, the carrier proteins, we end up with this balance between how many are free and how many are bound. And this is something that is studied as well, the level of free versus bound messenger in the tissue, and we'll see that again. So the diagram does a really good job of showing this. Most of it's going to be bound to carrier protein, but some of it will be free, and when you reach that target tissue, the free messengers will automatically cross, meaning the free hormones that aren't bound will cross into that tissue, go to the target cell, whereas that shifting in the concentration of the free messengers in the blood causes more of the hormones to be released, and that allows for further activation of the receptors. Once the hormones get to the tissue, they also encounter something called chaperone proteins. And these chaperone proteins help to cause the binding of the hormone to the receptors. Now, you've probably learned about heat shock proteins in a different way, but these are the same type of proteins that are used here. So they're also called heat shock proteins. That's why it's HSP90. And basically what they do is they keep the receptor ready to be bound. Once the hormone binds to them, they're released. That activates the receptor. It makes the receptor become the transcription factor that it is. It changes the shape of the receptor, releases certain parts of those heat shock proteins and the other parts of the receptor. The other thing that we'll talk about in a moment is you also usually have to have two receptors bound together. So that's shown here as the GR and the GR bound together in order for it to act as a transport protein. So again, we have chaperone proteins associated with these receptors that the hormones are binding to. Here we can see a nice general overview of how steroid hormones are activated. Again, we've got the free steroid hormone that's actually going to cross the membrane that kind of bounces back and forth between being bound to a plasma protein depending on where it is in the blood and the gradient, the concentration gradient of that protein. The free steroid then passes into the cell and it binds to a receptor. Now, it depends on the type of steroid hormone as well as the cell as to whether that receptor is in the cytoplasm or in the nucleus. In most of the literature, you will probably see these referred to as nuclear receptors because that's where they were first discovered, but we do find them in the cytoplasm as well. So the steroid hormone binds to the receptor and changes its shape. And we'll look in the next slide, but this also includes the incorporation of chaperone proteins because they help to maintain the shape of the receptor once it has that bound hormone. Okay, so I'm at step 4 now. You've got your steroid hormone bound to the receptor, and the receptor is activated. And what happens is it forms these things called zinc fingers, and the zinc fingers help to align the receptors on the DNA and cause transcription of messenger RNA. Now, the other thing to note here is that you always also see dimers forming. Now, they could be homodimers, meaning the same type of receptor binds together in order to get transcription, or they could be heterodimers, meaning there's two different receptors that bind together. But we always see these dimers forming. And then, of course, you get transcription and translation, as you can see in step 7 and 8. Now, this gives us a specific example using a progesterone receptor, and it incorporates those chaperone proteins that I mentioned previously and that we looked at previously. So, in the progesterone receptor, you get progesterone binding to the receptor itself, and the chaperone proteins kind of change shape and leave as the receptor undergoes phosphorylation. Then the receptors go into the nucleus, and notice now they have zinc fingers, and they form a homodimer. So two progesterone receptors come together. That allows for something called an adapter protein to bind to it. Oh, the other thing to note here is the HRE, or hormone response element. That's in that's what tells the progesterone receptors what to bind to. Then the adapter protein binds to the top of that, and then you have a formation of this apparatus that allows for transcription. So this diagram gives you a bit more detail about what the receptors bind to and to allow for transcription to occur. Here we can see the zinc fingers in diagrammatic form, and basically what this is once the chaperone proteins leave, the ligand binds, the chaperone proteins leave, there are these zinc ions that are attached to a series of amino acids that form these finger-like projections. And once those are exposed, they're able to bind to the binding sites, specifically the area in the hormone response element of the DNA, and then that binding allows for the transcription to occur. Here you can see a summary of the different receptor complexes that form depending on different hormones. And so in the top there we see several examples of heterodimers, and you could put thyroid hormone in there as well. And then in the bottom, notice that most of what you think of as steroid hormones, all of the sex steroid hormones, are homodimers. So again, what this is showing is the two pieces of the two receptors that have to bind together in order to cause changes in the DNA, to bind to the DNA. Here we have the thyroid hormones, and these are a type of steroid hormone that does have a thyroid binding protein that allows for the thyroid hormone to be transported in the blood. It is produced in the image to the left there. It's produced by the follicle cells using the colloid, the thyroglobulin, which we'll look at in a moment. And this diagram summarizes the chemical process that occurs to take your thyroglobulin, basically, that contains tyrosine, and turn it into the thyroid hormones. Now, I'm going to refer to them as T3 and T4. And we also have reverse T3 as well, so or RT3. I'm not going to use their chemical names. And something to know as we talk about this, so again, you end up with these class of hormones called thyroid hormones. The active hormone, the one that's actually going to do anything in the cells, is T3. But T4 plays a very important role in the transport and basically the stability of the thyroid hormone before it gets to the cell. So most of what's being transported around the body is T4, and then when it gets to whatever cell it's acting on, it gets turned into T3 or RT3, which again, all of these names are based off of where you find the iodine. So as you can see, T4 has 4 iodine, T3 and RT3 have 3, but the location of that iodine is what's important in their naming. All right, so this is a diagram from a different book, but I think it does a really good job of summarizing what we're going to look at in more detail. So you have inside that colloid, which is that kind of purple, pink stained area in the middle of the follicle cells, you have thyroglobulin. And in order to have the T3 and T4 metabolized, have them created, you need iodine, which is transported in blood as iodide. Then it's transported across the follicle cells in that stage 1 there into the thyroglobulin. Once in the colloid, the thyroglobulin and the iodine are combined. We're going to look at that in a moment. And we get endosized thyroglobulin that has that iodine attached to it. And then you get this general process shown there where you have enzymes that act on the thyroglobulin to turn it into T3 and T4. And then that is what goes out into the blood and is transported on the transport proteins, the thyroid transport proteins shown there in the blue boxes. So again, we're going to look at this in more detail, but this is a nice general overview of the steps. So here we can see more detail about what is occurring in that colloid, in that middle area where we have the thyroglobulin. And basically what thyroglobulin is, it's just this large, or not large, but long protein made up, or peptides that holds the tyrosines in place, so it gives correct structure and to where they're located next to each other. And then an enzyme called thyroid peroxidase comes in and activates the iodine, attaches the iodide, as you can see in B and C. So the thyroid peroxidase activates the iodide, it attaches it to the tyrosine, and then through the orientation of the thyroglobulin and the enzyme, the thyroid peroxidase, a phenyl group, or ring, is detached from one of the tyrosines and attached to the other one. And then this is the final product, the T4. And notice you have 4 iodides there. So that is what you have. And then that whole thing kind of gets taken into the cells, and then you have to detach the T4 from the thyroglobulin. And that's something that we'll talk about a little bit more later in the semester. Okay, so now we have the T4 and the T3, if one of the iodine has been removed, released from the follicles, goes into the blood, transported on a binding protein. And then when the thyroid hormones reach the cell that they are going to be active in, the T4 has to be turned into T3 in order for it to be able to be activated. And this is through a type of enzyme, a deiodinase, that takes the iodide off of the T4 to convert it to T3. And sometimes that doesn't go well, and you end up with RT3, which isn't an active form of the thyroid hormones, but we do see it in circulation sometimes. And then, we'll talk about the metabolic effects of this another time. So, getting into a little more detail about how the receptors are activated and what they do when they reach the DNA. You can see here you have T3. Again, this is saying it's T3, but we all know that it's more likely to be T4 that's converted into T3. So the T3 binds to a thyroid receptor, which are going to be in the nucleus. And then the thyroid receptor actually binds to another type of receptor. So this is a heterodimer. It's not a homodimer like what we looked at previously. And so the thyroid receptor binds to the RXR, the other receptor there, before we can have transcription. So again, you need this heterodimer. Notice that there isn't anything, there isn't like a ligand that binds to the RXR. It remains kind of empty. But they both have zinc fingers, they're both activated. And we also have a thyroid response element here as well. So in order for the heterodimer to be able to bind, you need them to kind of the zinc fingers to find that TRE, that thyroid response element. And that tells where on the DNA the transcription factors, the receptors should start reading. And then we get production of messenger RNA and new protein and a generic category of biological response. So again, this is an overview of how thyroid hormones work at their target cell. I do want to talk briefly about what happens to the thyroid hormones once they've been used or no longer needed in the body. And one thing, I mentioned the RT3, that is a metabolite of T4 that is more quickly removed from the body than the regular T3. So that's a useful way for the body to maintain levels of active versus inactive thyroid hormones. So what we see is that when we have lower levels of T3, we see a commensal lowering of the RT3, theoretically to then allow for an increase in the active compound of T3. So there does seem to be some feedback from how much RT3 and T3 there is. We also have these metabolites that are listed here, tetrac and triac, and then the others. Basically, these are, again, they're metabolites and they're very water soluble, so they're going to be removed from the body very, very quickly, and that will allow for the maintaining the levels of thyroid hormones in the blood, and not because some of these can bind to receptors and cause changes, you don't want them sticking around for a long time.